So as in the last video we have discussed about the act of maturation. Alright, so in today's video we are going to discuss about the formation of urine. Alright, so there are 3 steps of urine formation. So let me write here first of all urine formation. Urine formation. So there are 3 steps which will be followed to make urine formation. So let me make the arrows here so that I can write. Alright. As I told you, there are 3 steps which will be followed to make the urine. So let me write this with this color. Alright. So the first step we have which will be followed to make urine is glomerular filtration. Because we know that in the nephron filtration, reabsorption and secretion happens. So the first step is filtration. So let me write here glomerular filtration. Alright. The second step which will be done by the PCT and some will be done by the DCT as well, DCT and loop of Henle. So that is reabsorption. Alright. So that is tubular reabsorption. So let me write here tubular reabsorption. The last step which will be followed to make urine is tubular secretion, because the things which we don't need in the paratubular capillaries will be secreted out in the nephron so that it can pass out from the body. So secretion, tubular secretion. Alright, so these are the 3 steps which will be followed to make the urine. Alright, so now we are going to discuss the glomerular filtration in very deep. Alright, so let me write here, or let me draw the arrow. So we are going to discuss about the glomerular filtration, and we know that glomerular filtration is happening where? It is happening in the Bowman's capsule. Like filtration is happening where? It is happening from the glomerulus, the capillaries. Alright, and it is then filtrate is going where? It is going to the Bowman's capsule. So let me write here, let me draw here. So basically this is my Bowman's capsule. So let me write here down what it is. I should write this with white color. Alright. So this is what? This is Bowman's capsule. Bowman's capsule. This is Bowman's capsule. So the cells of Bowman's capsule are parietal epithelial, but these are modified. So when these are modified, these are known as podocytes. So let me write here or make here. So these are basically podocytes. These aren't podocytes because these aren't modified here in this stage, but these will be modified soon as I'm making. This is the layer of epithelium. These are all cells. These are all cells. So now I should make these modified cells. So these are modified here. As you can see here. These are all epithelial cells. Alright. And here this is completed. Alright. So I should write here down so that it can be clear to you what these are. These are podocytes. These are podocytes, which are the modified cells of the woman's capsule. Alright, these are podocytes. Now I should make here the glomerulus. I'll not make here glomerulus, but I'll make here only one capillary, because I want to show you people the filtration. So I am making with this color now, not with this but with this. Alright, so here I am making one capillary. This is one capillary. Alright, this is one capillary I do have. So I should write here, this is what, this is one capillary I have. Capillary. Alright, so the cells which are present in the capillary are squamous epithelial cells. Alright, so I should make here squamous epithelial cells. These are what? These are squamous epithelial. Squamous epithelial, alright. So I should make on the base as well. Alright, so here I have made squamous epithelial. So let me label it. These are what? These are squamous epithelial cells. Because in every capillary, squamous epithelial cells are present. Alright, so these are squamous epithelial cells. Now, the holes which are in the squamous epithelial cells are known as fenestration, by what it will get filtered. Alright, so here I have some holes. Alright, here I have some holes. These are known as what? Fenestrations. So let me write here down. Fenestration. Alright, by what it will get filtered. So now what do we have? We do have base membrane here as well on the base of the capillary. So I should make here this as well with this color. So here I have basement membrane. Here I have basement membrane. Alright, so now we do have a part here too, like we do have a space here too in the podocyte, and that space is known as what? Is known as split pores, by what the filtration will happen and will come to the woman's capsule. Alright, so let me write here down. It will come here by these. So these spaces are known as what? Split pores. So I'm writing here split pores. Split pores. Alright. So here one need to be focused is what? Here ultrafiltration happens. Now what is ultrafiltration? We do have two types of filtration. Alright. One is normal filtration and another is ultrafiltration. So ultrafiltration always happens under pressure. So here pressure will be generated, then filtration will be happened. So this is known as what? This is known as ultrafiltration. So let me write here ultra filtration, which is the filtration under pressure. Alright, so here filtrate will be coming. So this is basically what? This is filtrate. And ultrafiltration is what? It is filtration under pressure. the filtration which is done under pressure. That we'll be discussing as well, how it is done under pressure. So this I have is what filtrate. Now we need to know one thing, how much filtration pressure we do have. Like we do have net pressure in the glomerulus. Alright, so that net pressure is, wait, let me write this with the bright color. Yes, this is fine. So net filtration pressure we have, net filtration pressure we have, that is 10 millimeter of mercury, Hg. Alright, so this is the net pressure we do have in the glomerulus. So we'll be talking about that as well, how this net pressure is made. We'll be discussing about that as well. So now we need to know that in 1 minute how many heartbeats are being done. Alright, so let me make here, so I'm making here the heart. This is basically what, this is heart. I do have heart. Alright, and in 1 minute how many heartbeats are done? In 1 minute 72 heartbeats are done. Alright, 72 heartbeats. Alright, so how much cardiac output we do have? We do have 5 liter. Like we do have 5 liter blood in our body, we know that. So cardiac output is 5 liter as well, because we know that the blood we do have in the heart, it will push all the blood to the organs. So we do have cardiac output, that is also 5 liter. It is the same rate we do have blood. So let me I should make with this color now. So I do have here capillaries, not capillaries but arteries. Here basically it is going to the organ which is liver. So I am making here liver. Let me write here down. This is liver, and here I am going to make the kidneys, because we know that it is going to the kidneys as well. So these are known as what? These are known as kidneys. Kidneys. And this artery is known as renal artery. Renal artery. And this is known as hepatic artery. Hepatic artery. Alright. So the artery which is going to the liver is known as hepatic artery, and the artery which is going to the kidneys is known as renal artery. Alright, so now we need to know that how much cardiac output we were having. We were having 5 liter cardiac output. 5 liter, and it was known as cardiac output. Alright, so how much blood is going to the kidneys? Alright, there would be some specific amount which will be going to the kidneys as well. So how much like blood is going to the kidneys now? We need to know that. So in the kidneys, like what blood kidneys are receiving? They are receiving blood of 1100 to 1200 milliliter blood. Alright, which is going to the kidney. So around 1/5 of the cardiac output is received by the kidneys. So I should write this as well. Around 1/5 of the cardiac output is received by the kidney. Cardiac output. Alright, so this was the blood which was received by the kidney. So this was all for today. In the next video, we'll be going with the glomerular filtration rate, alright, and the pressure which are present in the glomerulus and the Bowman's capsule. We'll be going with that so that we can take out the net filtration pressure which is present there. Alright, so in the next video, we'll be going with that. This was all for today.